Hello, good morning everyone. Today I am here to present my research on assembling a workflow for assessing the genetic diversity of Munroniapinnata and comparative molecular docking with its substitute Andrographispaniculata. Let me begin by introducing the plant. Munroniapinnata, which is commonly known as Binkohomba, is a rare, therapeutically important plant which belongs to the family Meliaceae. It's a hardy perennial which grows up to 10 to 15 centimeters in height. And these plants have different morphological features according to their geographical location. The most prominent feature is the leaflet number, and in Sri Lanka it varies from 3 to 7. And let's take a look at why this plant is important. In Ayurveda and other traditional medicinal systems, this plant is heavily used to treat several diseases. Therefore, the annual requirement of this plant is considerably high, which can lead to overexploitation of this plant. Therefore, the selection of commercially desirable genotype is important to establish proper conservation and utilization strategies. For that, we can use DNA barcoding. It's a standard molecular biology method that uses specific regions of DNA to identify species diversity. The most commonly used universal chloroplast barcoding regions are RBCL, MATK, and tRNH-PSBA. Andrographispaniculata, which is commonly known as HeenBinkohomba, belongs to the family Acanthaceae. This is also used to treat several diseases. And in Sri Lanka, this is used as a substitute for Munroniapinnata because both plants are known to have similar pharmacological properties. And both these plants consist of bioactive natural compounds with therapeutic potential, which may be useful in pharmaceutical industry to develop new drugs. However, pharmacological properties of the phytocompounds are not well defined, because testing each and every compound to various drug targets under laboratory conditions take more time and it is expensive. Therefore, in silico studies with molecular docking can be used as a solution in order to reduce the complexity of laboratory experiments. Molecular docking is a computational technique that used to estimate the affinity of association between two molecules. For example, affinity of association between ligand and a protein. Ligand simply refers to a small molecule that forms a complex with a protein to serve a biological purpose. And this technique allows to examine how a molecule interacts at their protein binding site and rank them for prioritization in subsequent studies. And let's see why this research was needed. The genetic diversity of Munroniapinnata were not clearly identified, and the binding potential of phytocompounds to various drug targets were not well understood. Therefore, my original objective of this research was to get a comprehensive molecular and biochemical understanding of Munroniapinnata and compare their biological properties with its substitute Andrographispaniculata. And I have completed up to PCR optimization of DNA barcoding and were not able to continue my research due to pandemic situation. Therefore, I moved to an alternative study to find out the potential of phytocompounds present in both plants in inhibiting inflammation. Through that study, it was specifically aimed to find out the binding profile of different plant compounds to the COX-1 anti-inflammatory target and to evaluate drug likeness properties of the selected compounds. And let's move to the method and results of molecular characterization. As the first step, the plants which represent each leaflet number were collected from different geographical locations. Among them, the most diverse two samples, which were 3 leaflet type and the 7 leaflet type, were taken for the initial analysis. And from them, both petiole and leaf materials were taken for DNA extraction. And the DNA extraction was done using CTAP method. And according to the results obtained, the leaf materials that taken from each plant type were given better results compared to petiole samples. Therefore, they were selected as the suitable planting material for the DNA extraction. And then the extracted DNA was used for the primer optimization. And the best results could be obtained at 55 degrees Celsius. Therefore, it was selected as the suitable annealing temperature for the chosen primer tRNH-PSBA. And let's move to the method of the in silico study. As the first step, the selection of ligands and the receptor protein was done. And in order to prepare the receptor protein, the three-dimensional structure of the COX-1 complex was retrieved from the protein data bank. And then it was visualized using UCSF chimera. And the protein preparation was done using Autodoc tools. And the preparation of ligands was done using Autodoc tools. And redocking and validation was done using Autodoc tools and Autodoc Vena. And the selected compounds were docked to the target protein using Autodoc Vena in order to get their binding affinity values. And finally, the dock complexes were visualized using PyMol software. And after getting results, the compounds which had better binding affinity values were further evaluated using ADME prediction, target prediction, and toxicity prediction. And let's move to the results of the in silico study. As the first step, the list of phytocompounds present in both plants were selected from scientific literature. And these compounds were used for the docking analysis. And selection of receptor protein and the native ligand plays a crucial role in molecular docking. And in the case of inflammation, there are two main types of enzymes which involve in inflammation. They are cyclooxygenase 1 and cyclooxygenase 2. In the pharmaceutical industry, these cyclooxygenases are the most common target for anti-inflammatory drugs. Therefore, the COX-1 was selected as the target protein during this study. And aspirin, indomethacin, SC560, mofesolate-like drugs have been so far identified as COX-1 inhibitors. Therefore, during this study, the indomethacin was used as the positive control. Let's move to the preparation of protein. As you can see, the unprepared protein consists of large amount of unwanted inbound molecules. And these red color dots denote the water molecules. When prepared in protein, all these unwanted molecules were removed as they do not have any contribution to the binding. Similarly, water molecules also removed in order to ease the computation and clear the active site. And then hydrogen bonds were added to the system in order to stabilize the complex, and all missing atoms and residues were repaired and charges were added to give an optimum conditions. And this is the prepared protein that obtained after all these changes. And let's move to the redocking and validation. When we usually perform docking, the ligands have an ability to go and bind to anywhere at the target protein. Therefore, validation is necessary to ensure the ligands go and bind to the respective active site of the protein. For that, during this study, the co-crystalline ligand, which is indomethacin, was extracted from the protein complex and redocked to its receptor again. By that, the dimension of the active site was obtained. And in addition to that, in order for a ligand to serve as a therapeutic molecule, it should bind to the specific catalytic site with a significant binding affinity. The more negative the binding affinity value is, the more stronger the modulating effect. Therefore, the compounds which have more negative binding affinity values are predicted to have better therapeutic effect. Therefore, in order to compare the binding affinity value of the selected compound, the binding affinity value of the non-drug indomethacin was obtained, and it was minus 8.1 kilocalorie per mole. And these show the top 5 docking results of the selected compounds against COX-1 protein. As you can see, the campesterol from Munroniapinnata and 7-O-methylvagonin from Andrographispaniculata got better binding affinity values compared to the indomethacin. And this is the image that shows a binding pose of 7-O-methylvagonin, which is in red color, and campesterol, which is in yellow color, inside the binding cavity of COX-1 protein. And this is a superimposed image of a native ligand indomethacin and one of the core molecules 7-O-methylvagonin with their target protein. As you can see, the two molecules got overlapped each other, which gave us a further evidence that our core molecules also bind to the respective active site. Since campesterol and 7-O-methylvagonin got better binding affinity values, they were further evaluated using ADME prediction. It's an abbreviation for absorption, distribution, metabolism, and excretion. And this is designed to investigate how a chemical processed by a living organism. And Lipinski rule is the most commonly used rule to evaluate drug likeness properties of the selected compounds. It states that an orally active compound should not have more than 5 hydrogen bond donors, 10 hydrogen bond acceptors, the molecular weight should less than 500 Daltons, and the log P value should less than 5. And this is the drug likeness results of the selected ligands. And as you can see, the campesterol violated Lipinski rule by having more than 5 log P value. Therefore, it should modify before further analysis. And in the case of 7-methylvogonin, it obeyed Lipinski rule with no violation. Therefore, it was further evaluated for their target prediction. And these pie charts show the top 9 macromolecular target classes of 7-methylvogonin. As you can see, it has 8% probability to bind to the oxidoreductase class, which includes cyclooxygenase enzyme. And toxicology studies also often a part of drug discovery process. And results reveal that 7-methylvogonin did not violate most of the toxicity levels. However, further clinical trials are necessary to ensure their toxicity levels. And according to the results that obtained throughout the research, it can be concluded that both plants consist of phytocompounds that are binding to the COX-1 target protein with significant binding affinities. Among them, the best binding profile could be obtained from campesterol and 7-methylvogonin. Out of these two, 7-methylvogonin was predicted to have best ADME properties. Therefore, it's a promising compound to pursue further by using advanced computational methods and in vitro experiments to explore its potential against inflammation. And in this way, we can evaluate binding affinity values of different phytocompounds against other drug targets. And by that, we can sort out the most important phytocompounds against different drug targets and reduce the complexity of laboratory experiments. And these are my references. And finally, I would like to convey my sincere gratitude to all the people who helped me throughout this project. Thank you.